Welcome once again to Plant Physiology Bio 211D, Unit 1, Session 3, Photosynthetic Pigments and Light Absorption. By the end of this session, students should be able to list the pigments that capture light for photosynthesis, show the photosynthetic pigments in relation to the role play in photosynthesis, explain the color of vegetation in relation to light absorption, and you give an account of the absorption spectrum and the action spectrum. Pigments that capture light for photosynthesis plants and the algae are autotrophic, but this ability of theirs is made possible by the possession of pigments in the chloroplast. Now, what are pigments? Pigments are chemical compounds which reflect only certain wavelengths of visible light. This makes them appear colorful. Flowers, corals, and even animals can contain pigments which give them their colors. More important than their reflection of light is the ability of pigments to absorb certain wavelengths. Chlorophylls are greenish pigments which contain a porphyrin ring. This is a stable ring-shaped molecule around which electrons are free to migrate. Because the electrons move freely, the ring has the potential to gain or lose electrons easily, and thus the potential to provide energized electrons to other molecules. This is the fundamental process by which chlorophyll captures the energy of sunlight. There are several kinds of chlorophyll, the most important being chlorophyll A. This is the molecule which makes photosynthesis possible by passing its energized electrons onto molecules which will manufacture sugars. It is the most abundant pigment in plants. Chlorophyll A absorbs light with wavelengths of 430 nanometers, that is blue, and 662 nanometers, which is red. It reflects green light strongly, so it appears green to us. It has the composition C55H72O5N4Mg. All plants, algae, and cyanobacteria which photosynthesize contain chlorophyll A. The second kind of chlorophyll is chlorophyll B. Chlorophyll B has the composition C55H70O6N4Mg. It is different from chlorophyll A because it has a CHO group attachment instead of a methyl group. It exhibits a blue-green visual color. It absorbs light of 453 nanometers and 642 nanometers maximally. It is not as abundant as chlorophyll A. It helps increase the range of light a plant can use for energy. It occurs in all plants, green algae, and some prokaryotes. There is usually about half as much chlorophyll B as the chlorophyll A variety in plants. There's also bacterial chlorophyll A, which is different from chlorophyll A and chlorophyll B varieties by having a carbon-oxygen methyl group. The magnesium in the structure of chlorophyll is the reason why plants need soils rich in this mineral. A soil deficient in magnesium causes yellowing of the leaves. Carotenoids are usually red, orange, or yellow pigments and include the familiar compound carotene, which give carrots their color. Carotenoids are persistent than chlorophylls, and so during autumn, when the leaves lose their chlorophyll, give the leaves the pleasant reds, yellows, and oranges of the autumn foliage. Here on UCC campus, the Indian almond shows this characteristic in the dry season. You may look around to observe this phenomenon in some other plants during the dry season. These compounds are composed of two small 6-carbon rings connected by a chain of carbon atoms. As a result, they do not dissolve in water and must be attached to membranes within the cell. Carotenoids cannot transfer sunlight energy directly to the photosynthetic pathway, but must pass their absorbed energy to chlorophyll. For this reason, they are called accessory pigments. One very visible accessory pigment is fucoxanthin, the brown pigment which colors kelps and other brown algae as well as the diatoms. Lycopene, which gives the color of ripened tomato, is also a carotenoid. The range of light absorption by carotenoids in leaves is extended somewhat toward the middle of the visible spectrum. That is, carotenoids absorb light maximally between 460 nanometers and 550 nanometers and appear red, orange, or yellow to us. Pigments that capture light for photosynthesis. Phycobilins are water-soluble pigments and are therefore found in the cytoplasm or in the stroma of the chloroplast. They occur only in cyanobacteria and rhodophyta. Research implications of the phycobilins. Phycobilins are not only useful to the organisms which use them for soaking up light energy, they have also found use as research tools. Both phycocyanin and phycoerythrin fluoresce at a particular wavelength. That is, when they are exposed to strong light, they absorb the light energy and release it by emitting light of a very narrow range of wavelengths. The light produced by this fluorescence is so distinctive and reliable that phycobilins may be used as chemical tags. The pigments are chemically bonded to antibodies, which are then put into a solution of cells. When the solution is sprayed as a stream of fine droplets past a laser and computer sensor, a machine can identify whether the cells in the droplets have been tagged by the antibodies. This has found extensive use in cancer research for tagging tumor cells. The absorption spectrum. Sunlight is actually made up of different wavelengths of energy called the electromagnetic spectrum, part of which is composed of different colors which can be made visible on a screen when sunlight is passed through a glass prism. In nature, this is observed in the rainbow when light passes through water droplets, the droplets having the same effect as the glass prism. The colors displayed in the figure below actually represent different energy levels that constitute radiant energy, and it's as that's displayed. You have white light passing through the glass prism, and you have a range of light. You have red, orange, yellow, green, blue, indigo, and violet. So the light is split up into its component colors, and this tells us that the refractive index depends on the frequency of light. Violet has the biggest frequency. It is deviated the most. So the absorption spectrum. Now let us consider the substantive issue, the absorption spectrum. If you will quickly go over what we just discussed about the pigments, you'll notice that we made the important observation that without them, there would be no photosynthesis because they capture and provide the energy needed for the process. We also mentioned the various parts of the visible light each pigment is sensitive to and absorbs. Wouldn't you like to find out how it was determined to know the particular range of radiant energy each pigment absorbs? To find out the range of sensitivity for each pigment, chlorophylls A and B and carotenoids are exposed to white light in an instrument called spectrophotometer. It is found that chlorophylls A and B absorb light in the blue-violet range and the red-orange range. The chlorophylls do not absorb green light. Therefore, light of this color is reflected back into the atmosphere, which we see and therefore see that chlorophyll is green in color. When the wavelengths of the part of the visible light absorbed are measured and plotted on a graph, we get what is referred to as the absorption spectrum. So this is the absorption spectrum as displayed in this graph. Absorption by the wavelength. So we now have our proof of the range of radiant energy associated with each pigment. Come along and let us find out whether the range absorbed is what is used in photosynthesis. Let us refresh our memory with the summary equation of the process of photosynthesis. 6 molecules of carbon dioxide by 12 molecules of water plus light, giving us glucose plus 6 molecules of oxygen and 6 molecules of water. You notice that the gas released during photosynthesis is oxygen. This means that if we could create a system whereby oxygen can be connected and measured, whilst chloroplasts are absorbed to exposed to the particular ranges of radiant energy noted in our earlier discussion, then the oxygen collected can be used as a gauge to tell whether or not photosynthesis do take place. This is what the German physiologist T.W. Engelmann in 1881. He placed a filamentous green alga under the microscope and illuminated it with a tiny spectrum of visible light. In the medium surrounding the strands were motor aerobic bacteria. After a few minutes, the bacteria had congregated around the portions of the filament illuminated by red and blue light. Assuming that the bacteria were congregating in regions where oxygen was being evolved in photosynthesis, Engelmann concluded that red and blue light are the most effective colors for photosynthesis. With modern instruments, a lot of the rate of photosynthesis as a function of wavelength of light produces the graph shown in the figure. It is more precise than Engelmann's, but it tells the same story. It is called the action spectrum. So the rate of production of oxygen can be used as a means to measure the rate of photosynthesis at each wavelength of light. The action spectrum proves conclusively that the light absorbed by the chlorophyll contributes extensively to photosynthesis. The proof is made more evident when the absorption and action spectra are plotted on the same graph. The figure below shows that the peaks of photosynthesis correspond with the range of wavelength absorbed by chlorophyll A and chlorophyll B. So we have the absorption spectrum and the action spectrum. The absorbance is in red, whilst oxygen evolution rate is in blue. That's the action spectrum. So absorbance against wavelength, the visible spectrum. So in review, in this session, we have considered the meaning of what a pigment is and the role they play in absorbing radiant energy for photosynthesis. We have seen that sunlight, which is the same as visible light, lies midway between the high and low energy wavelengths of the electromagnetic spectrum. We also noted that all the photosynthetic pigments absorb only certain parts of the spectrum, specifically in the ranges of blue, violet for chlorophyll A and red, orange for chlorophyll B. The carotenoids are accessory pigments. The absorption spectrum shows the range of visible light that is utilized for photosynthesis, and the action spectrum gives conclusive evidence that the light absorbed is used for photosynthesis. The absorption spectrum shows how strongly the pigments absorb at different wavelengths. The absorption spectrum and action spectrum show that the wavelengths that are mostly strongly absorbed, red and blue, are the ones that cause photosynthesis to proceed at the fastest rate. Green is not only strongly absorbed, rather it is reflected, causing leaves to low.